Good day, panelists. We are the Group 7 from the Block 1, and we are going to present to you our research titled The Effectiveness of Goosegrass Ericin indica Leaves Extract as Primary Stain for Schaefer-Fulton Method. I am Gian Sinlao,
I am Angelianes.
I am Charlene Reyes.
and welcome to our presentation. We are going to study the important components which will um, contribute towards making the study a success. So these are the variables involved in the paradigm of the study. The diagram shows that the researchers will be gathering goosegrass and will undergo extraction and chemical analysis to obtain the needed substances and to determine the active constituents present in the material that made it compatible as a primary stain. So using different densities and concentrations of the obtained extract, the researchers will be able to determine the material's compatibility as a primary stain. So the diagram presents both the independent and the dependent variable. Under independent variables, we are going to take the different densities and amount of goosegrass extracts. First, the diluted hydrochloric acid plus 25% leaves extract, diluted hydrochloric acid plus 50% leaves extract, diluted hydrochloric acid plus 75% leaves extract. And the final variable for the independent is the 100% leaves extract, which malachite green will serve as a control variable. The dependent variable will be the measurement activity of the goosegrass leaves extract as a primary stain for the Schaefer-Fulton method in terms of pigmentation, cellular penetration, solubility, alkalinity, and volatility. Microscopic examination of the bacteria will also be performed. So after learning the variables, we are going to present to you the statement of the problem. This study aims to determine the potential and effectiveness of goosegrass Ericin indica extract as a primary stain for the Schaefer-Fulton method. Specifically, this seeks to find answers to the following questions. Number one, what are the active constituents found in goosegrass extract which are essential for effective primary staining using the Schaefer-Fulton endospore staining method? Second, is there a significant difference between goosegrass and the commercially made malachite green in terms of A, staining affinity, which involves pigmentation and cellular penetration, and letter B, chemical stability, which involves solubility, alkalinity, and volatility. So this study is guided by a null hypothesis. The goosegrass extract would be proven as a suitable non-toxic alternative and as a primary stain for Schaefer-Fulton method than the standard malachite green primary stain for endospore staining. So our hypothesis is that there is no significant difference between the effectiveness of goosegrass leaves extract and the commercially made malachite green as a primary stain for Schaefer-Fulton method. So next, we are going to present to you the experimental flowchart of our study. So first is the phase one, which involves the data gathering procedure. So this research is about using goosegrass, Ericin indica leaves extract as an alternative for the primary stain in Schaefer-Fulton method. For the researchers to test the efficacy of the said extract, cultivation of Bacillus cereus will be performed. So the next phase, phase two, involves the preparation of the PEMBA plates. So PEMBA plates or the polymyxin pyruvate egg yolk mantled bromothymol blue agar medium is used as a highly specific and sensitive medium for the isolation and enumeration of Bacillus cereus. So the researchers will prepare the PEMBA plates at the University of Pangasinan Medical Technology Laboratory. Next phase is the cultivation of Bacillus cereus. So for the cultivation of Bacillus cereus, they will be placed in the PEMBA plates, which is our primary plating medium. And then these plates will be incubated at 35 degrees Celsius for the bacterium to grow for 48 hours.
After phase 3, we'll proceed with our phase 4, which is the collection of Ericin indica leaves extract, where we will be gathering goosegrass in certain areas of the province. After gathering, we will wash the grass thoroughly with water to cleanse the remaining soil or dirt. Then we will chop the gathered leaves in small pieces, approximately into 20 millimeters in size. After that, we will prepare the materials needed for the decoction of the chopped goosegrass to get the extract. We will proceed to phase 5, which is the pigment extraction. We will gather the materials needed, like our mortar and pestle, and we will place the goosegrass leaves in a mortar. A small amount of ethanol will be added and it will be grinded by a pestle to release the pigments into solution. The addition of ethanol will be continued as necessary as well as extract in order to create a few milliliters of very dark extracted liquid, enough to be used for the following experiment. We will now proceed with our phase 6, which is the preparation of extract concentrations. where we will be using diluted hydrochloric acid with 25 leaves extract. Then we will use diluted hydrochloric acid with 50% leaves extract. Then diluted hydrochloric acid with 75% leaves extract. And then another solution where there is the 100% of the leaves extract.
So after phase 6 is phase 7, in which we will determine the extract's chemical stability and staining affinity based on its absorbance using spectrophotometric methods. The stability of the extract's staining affinity will be properly measured through detection of any sudden increase in absorbance after the first measurement of the independent variables. And in phase 8, microscopic evaluation of each slide will also be done for the assessment of the color intensity imparted by both the Eleusine indical leaves extracts and the malachite green to the endospore forming bacterium. So after all of this phase, we will proceed to the data analysis in which we will wrap up our study.
And this concludes our presentation for our research for the effectiveness of goosegrass leaves extract as primary stain for Streptococcus. And we are open for questions.